Hi everyone, my name is Schapira, and today I'll be presenting my project aimed at identifying structural chemistry data gaps in the protein data bank to aid machine learning. This work was conducted at the Structural Genomics Consortium at the University of Toronto as part of the Target 2035 initiative. This project was largely completed with ICM Pro, a software developed by MolSoft. Binding pockets are fundamental to how drugs work. They are three-dimensional cavities within protein structures where small molecules can bind, altering the activity of the protein and in many cases leading to therapeutic effects. Inside these pockets, chemical ligands form precise molecular interactions with amino acids, such as hydrogen bonds, electrostatic or hydrophobic contacts, and aromatic stacking. The Protein Data Bank, or PDB, is the reference repository where such data determined by X-ray crystallography or other experimental approaches are made publicly available. Consider the cancer-associated protein BCL2 and the drug venetoclax from PDB entry 600K. Here we can see the protein BCL2 in gray along with the molecule venetoclax in yellow. Venetoclax nestles tightly inside the yellow and red BCL2 pockets and engages in multiple intermolecular interactions. Here, we can see the protein residues surrounding the red pocket, along with the intermolecular interactions taking place. The three blue dots between the protein and ligand represent a hydrogen bond, while the other two dotted lines represent the pi stacking interactions between the ligand and protein and within the ligand itself. This combination of shape and chemical complementarity defines the drug's affinity and selectivity. In 2023, the Nobel Prize in Chemistry was awarded for AlphaFold, a major breakthrough in structural biology. AlphaFold predicts the 3D structure of proteins directly from their amino acid sequence, including potential binding pockets, with remarkable accuracy. This has transformed our field. Before AlphaFold, experimental determination of protein structures relied exclusively on time-consuming and resource-intensive methods like X-ray crystallography or cryo-EM. Now, researchers can access predicted structures for most proteins within hours. For drug discovery, this means structure-based drug design is no longer limited to the fraction of proteins that have experimental structures. In blue is a protein structure determined by X-ray crystallography. In red is the same protein structure predicted by AlphaFold. We can see that the AlphaFold structure is very similar to the experimentally determined structure. Here, we can even see that the B factor, a metric for the confidence of the structural model, is similar between the experimentally determined structure and the AlphaFold structure. But predicting protein structures is only the beginning. The next frontier is to predict, given the 3D structure of a binding pocket, which drug molecules will fit with high affinity and selectivity. Imagine starting from the amino acid sequence of a new disease-related protein, predicting its 3D structure, identifying its binding pockets, and then using an advanced machine learning model to computationally design the best candidate drugs, all before setting foot in a wet lab. That is the vision for the future of drug discovery. What drives progress in the accuracy of these predictive models is data. AlphaFold succeeded because it was trained on thousands of experimentally solved protein structures in the PDB. Similarly, machine learning models for drug design require large, diverse datasets of proteins with ligands bound to their pockets. The richer and more diverse the dataset, the better these models can learn patterns and generalize to new targets. The PDB contains tens of thousands of drug-like ligand-bound protein structures. These are invaluable for training, but it also contains thousands of apostructures where binding pockets are not occupied by ligands. These apopockets are useless to machine learning for drug design as they don't contain any ligand-protein relationship data. These missed opportunities become especially important when an apopocket looks very different from any holo ligand-bound pocket. If that apo site were experimentally characterized with a ligand, it could represent a completely new kind of binding environment. This would be exceptionally useful for machine learning as it would increase the diversity of the training data set and improve the model's ability to design molecules for any given protein. These unique apopockets are what we call data gaps. Machine learning models learn patterns from their training datasets. Given a binding site in a new protein of interest with no known ligand, chances to find a ligand-bound pocket in the PDB with no matching patterns are low, but increase if the pocket is divided in smaller subpockets. Our goal is to identify apo subpockets in the PDB that are structurally diverging from holo pockets. our primary focus is on human proteins in the PDB expressed in E. coli, as these can, in principle, be readily expressed, purified, and crystallized. The next step will be for experimentalists to find ligands and crystallize the protein-ligand complex, thereby enriching training dataset for machine learning. This project contributes to Target 2035, a global effort to develop chemical probes for every human protein. By highlighting unexplored subpockets, we can guide experimental teams towards the proteins and regions most likely to yield new structural and chemical insights. First, to create the data for holo pockets, the Plinder database was downloaded with over 400,000 protein-ligand complexes assembled from the PDB. To focus on biologically and pharmacologically relevant cases, I applied filters to the database. The database was restricted to human proteins expressed in E. coli. The structures also had to be solved to a resolution of 2.5 angstrom or lower. The property filters shown were applied to exclude very small ions, crystallization artifacts, or ligands too large or polymeric to be considered drug-like. Finally, the PDB entries were restricted to 2,000 residues in order to make sure the PDB entries could easily be processed by Molsoft's ICM. This reduced the dataset to a high-quality, relevant collection of 23,204 complexes. To create the data for the APO pockets, a code was run to send API requests for every chain of every PDB entry. These requests found the relevant information of each protein chain, creating a table with the PDB entry, chain value, ligand ID, UniProt ID, expression system, start residue, end residue, and origin. I then removed all entries containing ligands that were present in the holo dataset and filtered the table to only contain PDB entries with human proteins expressed in E. coli with a resolution of 2.5 angstrom or lower and a maximum chain residue length of 2,000. This left us with apoproteins that were structurally relevant to the data we wanted to find. For each structure in the holo dataset, I first isolated the binding pocket. I located the ligand, created a 10 angstrom sphere around it, and removed all atoms outside this region. I then used the ICM pocket finder algorithm to generate the surface representation of the pocket and removed any pocket that wasn't interacting with the ligand. This provided a clean 3D mesh that defines the cavity surrounding the ligand. For the APO dataset, I did not have a specific pocket in mind, so I ran the ICM pocket finder on the entire protein chain. I then removed all the pockets that were predicted to be undruggable based on their DLID, a druggability index that accounts for the volume, shape, and hydrophobicity of binding sites, and then found all the relevant pocket data for the pockets remaining. The next step was segmentation. Binding pockets are not uniform cavities. They contain subregions that ligands exploit differently. To identify these subpockets, I built a distance matrix of the pocket surface points and performed UPGMA clustering on these surface points. I then cut the dendrogram at 9 angstroms and was left with structurally accurate subpockets determined by the pocket surface points. I then colored each subpocket differently in order to display each segmentation of the cavity as distinct regions. Once segmented, I enriched these subpockets with chemical and structural annotations. For each surface point, I identified the nearest pharmacophore features within 5 angstroms. This was done by creating a sphere around my pocket surface point, then performing a consensus pH4 by APF with ICM. Here we can see the electron-positive and electron-negative pharmacophoric features. The rest are not shown as they would make the visuals too crowded. Once the pharmacophoric features are found, a distance matrix between each surface point and all the pharmacophoric features is computed. Then, the minimum distance can be found by taking the minimum value of each matrix. As we zoom into a specific point of the protein, we can see that the structure is colored based on the type of atom. In red are oxygen atoms. Once we show the H-bond acceptor pharmacophoric points, we can clearly see that the oxygen atoms in the protein have a pharma point around them. We can then relate this to the ligand. In blue are the H-bond donor points. The main one, in the middle of the screen, comes from a hydrogen bounded to a nitrogen. This creates a hydrogen bond donor, which, as you can see, is very close to the ligand's oxygen, which would serve as the hydrogen bond acceptor. This creates one of the points in which the ligand is connected to the protein. Another example of this is with the ligand's hydrogen, in gray, connected to a nitrogen, creating an H-bond donor. This forms an intermolecular bond with the protein's oxygen, which serves as an H bond acceptor. To obtain the data for the section of the protein around each pocket, a sphere of 4 angstrom was made around the pocket surface point. All the protein's atoms' XYZ positions were found, as well as their associated amino acid identity. Here, as you can see, the protein's atoms within 4 angstrom of the pocket are highlighted as shown. Sometimes, the amino acid numbering in the PDB does not match with the proper numbering obtained from the UniProt's reference protein sequence database. In order to fix this, both sequences need to be aligned. In both tables above, we can see myseq as well as the protein's name. The myseq column is the amino acid sequence from the structure, while the protein name column contains the downloaded sequence from UniProt. Getting the accurate UniProt sequence for the residues around the pocket is crucial, as it allows us to stay consistent with all sequences around all pockets, whereas if I used the PDB numbering, I would have to work around inconsistencies. When analyzing the atoms of the protein around each pocket, it is useful to know how far away each atom is from each subpocket to see how much they can affect the pocket's chemical features. I added a column to the table containing the distance from the surrounding amino acid atoms to the nearest subpocket surface points. This was done by creating a distance matrix between each surrounding atom and its correlated subpocket surface points. Then, finding the minimum value of the matrix will give us the distance to the nearest subpocket surface points. When ICM Pocket Finder creates a pocket, the surface points around the pocket are not evenly distributed, but rather distributed at each plane intersection. This causes regions where many points are very close together, which is unnecessary and causes redundant data. To remove these excess points, I created a distance matrix between all points of each subpocket. Then, I clustered these points using complete linkage hierarchical clustering. This means that points are merged into clusters based on the maximum distance between members, meaning that clusters only form if all points inside are within the set threshold of 0.3 angstrom. Then, the geological center of each cluster is computed, and only the point nearest to the center of the cluster is kept, and all the others are removed. As you can see, once the points are cleaned up, we are left with no more thick sections of surface points. I then map ligand atoms onto subpockets. Using a KD tree, each ligand atom was assigned to its nearest surface points. Ray marching determined burial depth, and weighted assignments were calculated. 50 rays were shot evenly around each ligand atom. Then, if any of these rays do not come within 1 angstrom of a surface point, then the atom would be characterized as outside the pocket. In many cases, the atom begins within 1 angstrom of a pocket surface point. In this case, the algorithm checks whether moving slightly outwards brings the ray closer to the surface. If so, that ray is considered blocked, suggesting the atom is enclosed. If the atom doesn't get closer, the ray is marched further out into space, and if it never intersects with the surface again, that ray is counted as escaping. After all rays are tested, the atom is considered inside the pocket if every ray is blocked, but even if some rays escape, the atom is classified as outside, with the escape fraction reported as its clearance ratio. In this case, the 6 atoms that are part of the aromatic ring are all characterized as outside, as well as the green and blue atoms connecting to the ring. Finally, the blue atom at the top left of the display, bound to a hydrogen, was also found to be outside the pocket. The atom furthest to the left is the one with the lowest clearance ratio. As you can see, the atom is barely outside the pocket, correlating with the clearance ratio. Once the algorithm has decided that an atom is inside the pocket, it assigns that atom to 1 or more subpockets by looking at the nearby surface points. It does this by finding the closest group of surface points to the atom with a KD tree search. Each of those surface points already belongs to a defined subpocket, so the algorithm tallies which subpockets appear most often among the nearest points. The atom is then assigned according to that distribution. For example, if all of its closest surface points belong to one subpocket, the atom is placed there. But if the nearby points are split between several subpockets, it records a percentage breakdown that reflects how much the atom overlaps with each one. As you can see, the final 2 columns give the atom subpocket assignment as well as their clearance ratio. Finally, in order to find duplicate pockets, all pockets coming from the same protein were grouped together. If the surrounding residues of a pocket were shared at least 50% with another pocket, then the pockets were characterized as duplicates. This was an important step, as oftentimes pockets from different PDB entries are the same, as the protein chain is similar enough to produce the same pocket. In this case, both the blue and red pockets come from different PDB codes. When we look at them superimposed, we can see how the pockets are really duplicates. Now let's turn to the results. In total, 5,216 unique holo pockets were found, as well as 16,084 duplicate ones. For the APO pockets, 53,700 pockets were duplicates and only 8,213 pockets were unique. The average pocket volume of the holo and APO pockets were around 450 and 375 cubic angstrom respectively. In terms of DLID, pocket druggability index, the holo pockets averaged around 0.4, whereas the APO pockets averaged at around 0.25. As you can see, the distributions of the holo pockets was significantly wider than the APO pockets, as the APO pockets with a DLID of under -0.5 were ignored. When comparing pocket volume with number of atoms per ligand, we can clearly see a relationship, showing that the larger the ligand, the larger the associated pocket volume. In terms of number of subpocket per pocket, we can see that both holo and APO pockets are very similar, both with around 1.8 subpockets per pocket. This number may be surprisingly low to some, and the reason is because many pockets are quite small enough that they only contain one subpocket per pocket. Finally, the average pharmacophoric feature distances range largely depending on the feature. As shown, the average distance for the positive and negative pharma points are way larger than the other four features, at around 6 and 5.25 angstrom respectively. The range of these distances is large, however, with their largest recorded distances being around 14.5 and 13 angstrom respectively. With respect to the hydrogen bond acceptors and donors, their average distances are around 3 and 2.5 angstrom respectively, which is in line with the distance separating hydrogen bonded heavy atoms, while their maximum recorded distances are around 9 and 6.5 angstrom respectively, indicating atoms that are not hydrogen bonded. While the aromatic and lipophilic values are similar to that of the donors and acceptors, the lipophilic has the smallest recorded distances, as they are the most abundant. Their averages are around 3 and 2.25 angstrom, while their maximum values are around 8.25 and 5.25 angstrom, respectively. With both holo and APO datasets now complete and already filtered to human proteins expressed in E. coli, the next step is direct comparison. Our goal is to identify APO pockets that are unique, pockets not represented in the holo dataset. These unique APO pockets represent data gaps in the PDB. Prioritizing them will guide experimental targeting under the Target 2035 initiative, leading to richer training datasets for machine learning. In this work, I provided a structured datasets of subpockets annotated with chemical and structural features that may be exploited as training set. Additionally, this work supports the experimental goals of Target 2035. By flagging underrepresented or unique APO subpockets, we guide experimental teams towards the protein and binding environments most likely to expand the PDB and enrich ML training. To conclude, this project demonstrated that subpocket segmentation is a powerful tool for bridging APO and holo structures, identifying data gaps in the PDB, and supporting both machine learning and experimental drug discovery. By highlighting where data is missing, we can better focus our resources on filling those gaps, ultimately advancing the mission of Target 2035. Thank you to the Structural Genomics Consortium and my supervisor, Luca Chiesa, for their guidance, and thank you for joining me today.